This is a study again from that project. This actually encompasses data from multiple states, including Frank's work here in Maine and some of the work I did in Michigan, where we looked at those summer morphs, so they're normal spotted-wing Drosophila that we see in our traps and expect, and those winter morphs, those darker ones that are that are coming later in the season. And we were looking at the overwintering survival, comparing those two different morphotypes. What it shows is those winter morphs are, these winter morphs are here in blue. They are better at surviving in in the blue is the summer morph, so they die off pretty fast. But the red ones, the winter morphs, they're able to survive for longer at colder temperatures, and they can basically survive for quite a long time. In this graph here on the right, you can see days out to at 0 degrees Celsius, so 34 degrees Fahrenheit. They can live for 34 days in those colder temperatures. So they can survive the winter. And you would picture when we have a nice, even when we have a nice cold winter here in Maine, if we have snow cover all the way throughout the year, underneath that snow where they're overwintering in the leaf litter, they're usually pretty safe and they can survive for quite a bit. Obviously this year we haven't had much snow cover, but it hasn't been too cold. So I think a lot of people are probably very happy to see the snow cover coming over the next few days to help save our bushes from winter injury. Okay, so that's the overwintering survival. So we know they move up and down the coast, which makes us wonder about insecticide resistance spreading around. Thankfully, we're not seeing too much spreading between the East Coast and West Coast, given that they have those resistant populations on the West Coast. But there is some movement of them going on. So when I talked with growers recently at the Ag Trade Show, I asked them what kind of information they'd like to see, what more information they'd like to see. And one thing was more information on trapping. So all of our information on how to trap and stuff is on the wild blueberry website, the UMaine wild blueberry website. But here in the lab, we trap with these little red solo cups. We use these stakes, which are stakes for flower pots, and we put them at the edge of the field. Why we put them at the edge of the field, I'm going to cover in a minute. And in these, we put a bit of yeast, a teaspoon of yeast, 4 teaspoons of sugar, and we put 12 ounces of water. Well, we mix that up. That's for tree traps. So we divvy that out across tree traps. And this works really well. This is what we've developed a lot of our thresholds on, which we'll cover in a minute. I put up this comment here because I know a lot of people still use apple cider vinegar, and that that works too. I would say that people should not discriminate as long as you keep trapping. So monitoring on your own farm is key for knowing when to manage spotted-wing Drosophila, or when you don't have to manage spotted-wing Drosophila, when your populations are really low. So thinking about our thresholds, so we developed, my predecessor, Frank Drummond, developed some really interesting thresholds based on those red solo cup traps. So we encourage people to trap on their farm, to hang up tree traps along the wood line, and based on the average number of males, remember those little fellows with the dots in their wings, you would either initiate your spray program, or you'd initiate harvesting based on the number of males caught across those tree traps. So a general rule of thumb is 3.5 males in the trap would mean a 90% probability of not having infestation the following week. So if you had 3.5 males, you have a 90% infestation or probability of not having infestation the following week. But that does leave you having a 10% probability of having infestation the following week. But the key take home would be to use these thresholds to trap on your own farm and to know basically from your local area what's going on, so you can inform your management based on that. Let's see, why is this not moving on? So we did assess these thresholds this year, we were monitoring at 10 sites across the mid-coast region this year. This is just a quick table showing when we first trapped SWD this year. So we trapped them a little bit earlier. As I said, I think it was average the 18th. It was one site that was a little bit later than all the others. We did find infestation pretty much at all sites that we monitored infestation at. And we did, and really the probability reflected what we found in the traps. So it was a good year to monitor our traps and check our thresholds, because every site that we would have estimated infestation would be found, we found it at. But there was one site, obviously, here where we didn't trap any males, but we also did find fruit infestation. And I think that's more of a function of the fact that our population of spotted was increasing so fast that they got ahead of us on that one. But for the most part, the thresholds did continue to work, that where we had those high male trap captures and the high probability of having infestation in the fruit, we did have that infestation. One thing my lab is hoping to work on this coming season is to look at one of these new different trap types for spotted Drosophila. I think a lot of people who might use the yeast and sugar trap type were probably always complaining that, oh, it's very smelly, it's sticky. I've got some feedback from growers that they just don't like doing it. So traditionally, a lot of people who use these liquid traps, these pictures from Cesar Rodriguez in New Jersey, they use just a clear trap with a clear drowning solution and one of those commercial lures that you can buy availably commercially. Another thing that's been shown to work pretty well is these red panel traps. So these are sticky cards like we monitor blueberry maggot with. What you do is you'd hang them from, a stake on the side of your bush. Here, this is a high bush bush, but we can hang them on the edge of our crop again in the shade, and they've hung a commercial lure on there. In their early data, you can see here on the right, these red panels were really attractive to spotted Drosophila. So they trapped a lot more compared to any other colored panel. So that's really interesting to see. And if you see in the bottom of this bottom graph here, the red panel and the lure caught more than the deli cup and the yeast bait. So what we probably have here. So I'm interested in testing these red panels to see how they shape up versus our red solo cup and our yeast solution, just to see if they're easier to monitor spotted Drosophila with them. You could basically have your red panel, go out, put it up, leave it out there, and you could go over and check it. It could stay in the field and you could scrape off flies as you wish, like a lot of people probably do with the blueberry maggot traps. Comparing the liquid yeast baited or liquid baited trap with the red panel trap, still though the red panel trap did work a little bit better here in New Jersey. But it is one thing that we're going to hopefully look at this summer in my lab. We don't recommend that you just do adult trapping. So a great way of knowing if your traps are working and your spray program, or whether you're harvesting quick enough for spotted Drosophila, is to do fruit sampling. So there is a way of actually sampling the fruit to look for spotted Drosophila infestation. So simply, you take some fruit, you would put it in a little container, and then you can bring it back to your house or your truck or wherever, and put it into a Ziploc bag with a strong salt solution. So what we mean by a strong salt solution is you take a gallon of water in a little gallon jug, you put in a cup of salt, give that a shake together, and you pour that in on top of your fruit. It says here to let it sit for 30 minutes. We usually let it sit for an hour, usually as long as it takes, or as long as it takes for us to get back to it. I tell people to go out, rake your blueberries in the morning, put it into a Ziploc bag, cover it up with your salt solution, maybe have your cup of coffee and your toast, and then maybe you could return to it after you've finished your breakfast. How we do it in the lab here is we took a funnel and we just chopped off the bottom of the funnel. We put a bit of hardware cloth, a bit of 1/4-inch hardware cloth in there, and we use a coffee filter like you can buy in Hannaford's or Walmart. It's one of those reusable coffee filters. I don't recommend using the same one that you've just made your coffee with. Use a separate one. And we'd pour the fruit and the salt solution into the upper container, and that flows away basically. And what's left in here is usually your larvae. It does work for blueberry maggot and spotted-winged Drosophila, but you can use that basically as a quick way to monitor your spotted-winged Drosophila or your fruit fly populations and make sure you're not getting infestation in your fruit. It doesn't take long. It takes about an hour. You can usually see, I think this was just with a regular camera lens, so if you have big larvae in there, you'll see them. But if you have a hand lens, you'll be able to see some of the smaller ones as well. So I mentioned monitoring and putting up traps, and I always recommend that you put up your traps at the edge of the field. One of the reasons we do that is to keep the trap out of the direct sunlight, in the shade of the tall trees at the edge of your field. It's also easier to access, but we also know that spotted-winged Drosophila are really active in and around that edge of the field and out in the woodlot. So the graph here on the right actually shows some data for blueberry maggot, which my predecessor developed, and Judy, who I think is on here. And they found basically that there is this strong edge effect with blueberry maggot fly. They hang out around the edge, and most of the infestation is at the edge. This graph is showing trap captures. So you can see the trap capture here at the edge is pretty high, and then it tails off. So they do hang out around the edge. And we've been investigating where spotted-winged Drosophila are hanging out and where we're seeing their infestation going forward here in Maine. So we've been setting out these gridded transects and taking samples in transects into the field to find basically if there's a pattern for where they're laying their eggs. We use that same salt flotation test to float the larvae and count them after that. So I'm going to show you some data from 3 sites today. The first site, I'm going to show you a time course where we sampled for 4 weeks every week, and we'll see basically how the infestation changed in the field. And then for the other 2 sites, I'm just going to show you the final snapshot, the infestation sampling we took right before harvest in an unharvested field, a managed field, and a holdover field. So this first one is the unharvested field. This was the very first day we sampled on the 23rd of July. And you can see really there wasn't much of a difference in color across these. So to walk you through it a little bit, the darker red it is, the higher infestation. The bluer it is, the lower infestation. So first day it was a small bit of infestation. Oh. There it is. My computer's freezing a little bit here. This is the second sampling day. There's not much change there. You can see here in the 3rd week, as we were getting into August and our populations of our adults were increasing a bit, we started to see a little bit of infestation creeping in towards the edge of the field. So it was really concentrated within those first 40 feet of the edge. And then in our final sampling, you can still see there's a huge concentration of spotted-winged Drosophila infestation towards the edge of the field. And you also had this bit of a swale here on the right-hand side. To explain that, it was actually because we had at this field, there was a large area of weeds off to the right of where we were sampling. So we've observed, and we've always known from blueberry maggot fly, that fruit flies will use weedy areas to, you know, work their way into the field, or they'll concentrate around these weedy areas. So when you think about your spotted-wing Drosophila and your fruit fly management, having good adequate weed control will help you out a little bit on that. Okay, so I highlighted this little red thing here because that's really a good indication of our infestation levels at our field. So darker red, the higher the number you see here in this, would mean that they had higher pressure. There was more spotted-wing Drosophila at that site. We're going to move on to our second site. So this was, oh, this should say holdover.
Hey,
so this is our holdover site.
Hey, Phil, there's a quick question.
Oh yeah, go for it.
Could you define the axes again? I think people are a little confused.
The axes here. So the axes on the bottom here is basically, we moved into the middle of the field and we ran 3 transects into the field. So this would be basically, you know, if you were at your woodlot, we'd done it across 300 feet of the edge. And then we sampled up to 160 feet into the field itself. So this would be the distance into the field, and this is just the distance along the edge of the woodlot. Hopefully that helps. Okay, so this was site 2. This should say holdover up here. I forgot to change that. So basically the trend is that even in the holdover field, we saw that there was this large edge effect in terms of infestation. There was a large concentration right along the edge. And let's see, this is the third field. This was a harvested field. So while this number in our previous 2 sites was 25 and 14, you can see this is a really low number here. It's 0.6. So they had a really low infestation here, mostly because they had sprayed and they'd actively managed for spotted-wing Drosophila. And again, there was a bit of an edge effect. There was a little bit of an inlet here. I'm not sure why that was, because this site also didn't have the weeds. This was just a regular site and had good weed control. So we see there was a bit of a weird pattern here, but really the infestation was really low at this site by the time we harvested. I think they were just about to harvest and they protected up to then. So obviously their chemical control strategies had worked pretty well at controlling spotted-wing Drosophila at this site. All right, so I'm going to try and move on to the next slide. It seems my computer is having some hiccups. It's not liking all of this presentation. So talking about current spray recommendations, I took the information from our chemical control insecticide sheet, and I put them into this little table here. So for conventional, we have a few different options. You'll see the chemical class here on the left-hand column, the insecticide here in the right-hand column, and you'll see a figure or a number, and that's basically the rating that we've ranked these insecticides on. So 0 would be not effective, 1 would be slightly effective, all the way up to very effective with 4. So there's a lot of good options for spotted-wing Drosophila control. I always put in this little blurb to obviously consult with your whoever you're selling your fruit to make sure that what you're spraying is on their approved spray list or it meets their standards for what they want your fruit. Obviously check with your pre-harvest intervals. If you've data for MRLs, just make sure you're spraying what's good for your market, basically. So thinking of these, I've colored them mostly to show how closely related they are. So different colors would be different chemical classes, and you'd want to rotate between those colors to stave off insecticide resistance, like I was discussing that they've already seen in California. I've made the neonicotinoids and the spinosins a pretty similar color because they really work on the same mode of action of insects. So they can be pretty similar in terms of their mode of action. So there's many different options. I won't go through them for brevity. And the organic side, obviously, Entrust is still one of our main insecticides. And that's a bit of an issue because it means that we can't rotate our chemical classes as effectively as in conventional management. And it is one of the reasons why they've had increases in resistance in California, because a lot of their acreage there was organically managed. So we are monitoring for insecticides or the susceptibility of populations to insecticides here in Maine. I don't like using the resistance word because we don't have resistance to insecticides here in Maine. But we have been monitoring. We started this year. In California, they have mostly seen resistance to spinosad, some pyrethroids as well, albeit they haven't really reported fully on that yet. So in California, they have this figure here. So this is 152.6 milligrams per kilogram in terms of their LC50 value. So that's the concentration that it would take to kill 50% of the spotted-winged Drosophila that they test for this susceptibility to the bioassays. So in Maine, we've done it here at 3 sites. We only fit it in 3 sites. We tested malathion and didn't find any problems. We also tested Entrust, and we did notice that there was some difference between our sites for Entrust. So we had 3 sites. Site number 1 here is pretty low. This was actually my back garden. We wanted a site where we had no previous insecticide sprayed. Site 2 was from an area that was minimally managed. I think they sprayed once. And site 3 was from a more heavily managed area. So what this shows is there was a little bit of a difference in susceptibility between our sites. Obviously, we're looking here at, I think it's around 37 milligrams per kilogram in terms of an LC50 value. So when you think about your California population, we're really way off. California were already seeing that they were spraying and they were still getting infestation in their fruit. So they were having some real problems. We're not having that here, but it does highlight that we need to rotate our chemical classes. So we need to make sure that we're not spraying the same insecticide consecutively within seasons. Another thing we're hoping to work on this year is an attract and kill spray. It's a bait spray for spotted-winged Drosophila. I just pulled this slide from Cesar Rodriguez. We have no data on it here in Maine yet, but it is something we're going to look at, where across a couple of weeks here in blueberries in New Jersey, they had a 77% reduction with this hook versus no treatment. And in the second week, they had a 67% reduction. So this is an interesting product. It's not one that you spray across your entire crop. You put little dots of it. You can see it here. You put a little dot of it, in different areas, and you could even maybe put it right at the edge, and it releases a little pheromone, and it attracts the spotted-winged Drosophila in. And when they come into it, they land on it, and they come in contact with an insecticide, and they die basically. So this data was from blueberries in New Jersey. I think this is from California, from raspberries. So I'm not really highlighting that as much, but they also saw a good reduction. We had hoped to test it this year, but we couldn't get our hands on it due to coronavirus. So that was a bit of an issue. All right, for the next little bit of the talk, I'm going to talk about biological control for spotted-winged Drosophila. The new project we just had funded has a big component where we're going to look for biological control. We're going to look at releasing biological control agents. I see I'm going to run over on time if I don't speed up, so I'm going to speed up a little bit. So this work was done by Kent Dana. You'll see his picture up on the right-hand side, and all of these people on the slide. And basically, over the last 4 years, we've looked for lots of different biological control agents here in the United States, native endemic parasitoids that might help us control spotted-winged Drosophila. What we found is there are many different parasitoids. Some will parasitize spotted-winged Drosophila, but others don't. So these two here on the left are pupal parasitoids. They do parasitoid spotted-winged Drosophila, but usually parasitism is pretty low, 0 to 10%. So not really effective at as a biological control agent to reduce the populations of spotted-winged Drosophila. Larval parasitoids, which would be more preferred because they'd attack the larvae and stop them developing further and damaging fruit. We have found lots of different species, but they attack all other Drosophilids, but they're not very effective at controlling spotted-winged Drosophila. One of the reasons we see that is because they get encapsulated. So when these native United States of America Drosophila parasitoids try to lay their eggs into spotted-winged Drosophila, spotted-winged Drosophila can encapsulate those eggs and stop it developing. So they're not effective at controlling spotted-winged Drosophila. They do try to oviposit into them, but they don't survive any further than that. So because we don't have effective biological control agent, Kent and his team, which are all pictured here, went out to China and Korea, where we think spotted-winged Drosophila was introduced in from, and basically searched for parasitoids in those areas that we could import here into the United States and release to control spotted-winged Drosophila. They found 3 really interesting and effective parasitoids. This one, Ganaspis brasiliensis, is probably our most promising hope. There's a Leptopolina that probably won't be released, and this, this Leptopolina, and then we have this Acebara species. And these are all been the ones that were brought to the United States for testing to potentially be released here. Ganaspis is probably our best hope at the moment. It meets a lot of the criteria, and it looks like it's pretty effective. This data is from their collections in China and Korea. They were looking at 4 different species that are common across those areas. 2 rupus, a strawberry type thing, and this scambus species, which is basically like elderberry. What you'll see in red will be basically the amount that had Ganaspis brasiliensis in them, or the amount of Ganaspis brasiliensis. Blue would be Leptopolina, that little Leptopolina that I mentioned there, and then yellow would be others. So the main take home for this is Ganaspis was found in a lot of the samples they tested. It was really good parasitoid, so they found it in most of the samples. And in terms of parasitism, it was around 20% in a lot of the different species, but it all went all the way up to 60% in some species that they were pulling fruit off of. So this was pretty promising. If we could get a parasitoid to basically take away 60% of our spotted-winged Drosophila and reduce the population, that would go a long way to help us controlling spotted-winged Drosophila. So a little bit about the background and the hoops that we have to jump through to get spotted-winged Drosophila released. So there's many different steps. You got to basically go out and find it. We've done that. You have to bring it back to the United States and do a whole load of experiments in a laboratory to make sure that if we release it, it's not going to decimate fruit fly populations here in the United States. For the most part, most people don't like fruit flies anyway, but they still make us jump through that hoop. And then it has to go through all of these different approvals through the different petitions for release. So this is where it's how it's been done. A petition has been put in for release. It's been checked by a few different places. They've done all of the host specificity testing and all of the fun stuff that's required to go through the release or the permit. And then it goes out to a commission that evaluates all of that data. One of the final steps of the petitioning process is it goes through tribal council. So the tribal council meets to approve its release. We heard this week that that just ended. It ended this week. So it's finally moving on to what we call the public comment period. So this is where growers and grower institutions and researchers and everyone can write to the government to basically say that we would really need this to be released, that it would be very beneficial that if it was released. So that's coming up soon, and we're hopeful that very soon we'll be able to release it here in Maine. We'll be rearing it in the lab, in my lab, and we'll be releasing it at sites up and down the coast in wild blueberry and other sites to hopefully affect our spotted-winged Drosophila population. So I'm going to end on this one. This is another thing to mention. It's a little interactive management support tool that was developed under that previous project by Miguel Gomez in Cornell University. And you'll probably, mostly of you will recognize this guy's smiling face. That's Frank Drummond. So my predecessor and Miguel worked on this. And it is to help growers decide when they should spray or when they should harvest based on a number of different metrics. So obviously early harvest works really well when our crop is very close to ripeness and our trap numbers are keeping up and we have the labor to do so. And they basically took a whole load of data from trapping and infestation, the economics of the crop, looking at what we'd expect in terms of our return and how much we spend on a spray for spotted-winged Drosophila. And they put it together into a little Excel sheet. So this Excel sheet, obviously, we have Microsoft Excel on most of our computers. And you can download this Excel sheet from the University of Maine's Wild Blueberry website. So here I actually just took a screen grab of my browser where I Googled University or UMaine Blueberry SWD Economics. And the very top search here is the one that will basically download this Excel file to your computer. The second is actually our spotted-winged Drosophila fact sheet, which we're always updating with new information. So that's another good one to look at while you're on the website. So I'm not going to open it up today and go through it, albeit we can potentially look for it if we have time. But basically, when you open it up, you'll see a number of different panels. The main one you need to be looking at is one that's this color here. It's kind of a yellow with orange dots. That's where you fill in all the information. So you fill in what kind of a type of farmer you are. You might be a low input one. You might be a high input. Your expected yields, the price you're expecting for your blueberries, which is always, a moving target, the amount of acres you have, how much it costs to spray, and basically it'll give you an estimation on what you might potentially be thinking of doing. Based on the economics of your crop, it'll tell you should harvest early, you should spray, etc. So it's a really effective tool. I'm bringing it up mostly because we're always trying to get feedback on these tools. So if anyone's using it, or if you plan on using it and you want to give us some feedback, please let us know. It's not a silver bullet. You need to continually also be monitoring and checking your infestation to be sure, but it can definitely help out with you making decisions on your farm. All right, so I'm going to end there. I'll talk a little bit about conclusions. So moving forward to 2021, we've had a bit of a mild winter so far. We don't know really what our spotted-wing Drosophila populations will look like this year, but I guess the recommendation stands that you should monitor early. Monitoring on your own farm is key. So getting your own data, getting your traps out is important. If you're doing consecutive spotted-wing Drosophila applications in a season, make sure you rotate your chemistry. So don't just spray, your delegate or your malathion or whatever multiple times in the same season, because that could lead to resistance, and we need to be conscious of resistance developing in our populations. I guess the silver lining is hopefully we might see some biological control agents being released here pretty soon for spotted-wing Drosophila if all goes well. With that, I'll take questions if anyone has any, if I haven't put you all to sleep. Check the chat.
There's one in the chat. As people think of more, feel free to type them in or shout them out. This first question is, does wet or dry affect their presence? So I'm assuming wet or dry season. And do you mind repeating the part about the salt test for the larvae?
Yeah, kind of course. So spotted-wing Drosophila they really prefer damp conditions. So while we had a pretty dry year here in Maine this year, we had basically no rain. It was still pretty humid, and it's the humidity is the one to look at. So if it's really humid, their populations will increase pretty fast. Again, if it's raining, that will drive up the humidity. Here we didn't have rain this year, which meant we were really looking for rain, but we still had high humidity throughout the season. So yes, if it's wetter, it's better for them. It's bad for us, of course. We'd like it rainy, lots of rain, but it to be drier in terms of the atmosphere, so spotted-wing Drosophila numbers don't increase. And yeah, let me roll back the slide deck here to go through the infestation data, or how to monitor infestation. Too far. So to go through infestation monitoring again, basically I'd go out, rake your blueberries. What we do here is we rake and we pour them into a small little cup. We only collect 8 ounces. We always collect it around the edge of the field because we've seen this trend that they're hanging out around the edge. We're currently not recommending border or perimeter management for spotted-wing Drosophila, but it does look pretty interesting. We'd like a few more years data and actually to do a few trials on that. But anyway, well, we collect our sample at the edge. We pour it into a little cup. We bring it back to, in our case, we bring it back to the lab where we've made up a strong salt solution. So we put, for every gallon of water, we put in 1 cup of salt. So it's a lot of salt throughout the season with all of our tests. We would put our sample into a bag and we'd cover it with salt water. So we put in plenty of salt water to cover the fruit all the way to the top. And then we leave it in there. It says 30 minutes here, but I would leave it about an hour just to be safe. What that does is that salt solution causes any infestation or any fruit flies you have to wriggle out into the salt. Obviously, hopefully you don't have any wriggling out into your salt, but if there's anything in there, it should wriggle out. Then what we set up is this kind of 2-filter system. So we took a regular funnel that we bought. We chopped the bottom off it with a hacksaw, and we put in a little bit of hardware cloth to catch the fruit, but allow the fruit flies to fall through. So we pour our strong salt solution onto that hardware cloth, and under we have this coffee filter to catch the larvae going into it. And then we would basically just use a microscope in the lab because we have them, but a strong hand lens would be just as good, and we look for the larvae. If you go onto our website, the Wild Blueberry website in UMaine, you'll be able to download some information on this that really focuses in on what the larvae look like. I didn't include it in this presentation, but these white squiggly larvae here is what you're looking for. It's not what you want, but if you find them, you'll know basically that you have some infestation going on. We recommend that you check it regularly to know that your spray program is working, or to know maybe you need to avoid that area of the field and maybe harvest somewhere else, etc. So hopefully that answered the question.
Yeah, I think so. And I also put, you guys have a great video of the SWD trapping, so I put that link in the chat.
Okay. Since the model for trapping have different degree of effectiveness, how can we set a universal threshold triggering sprays? Yeah, so obviously we want to have a threshold that's low enough that we don't get infestation. Obviously your processor or your consumer is going to be, they might find infestation and they're probably not going to want that. So you want your threshold to reflect your market. So obviously, if you're going for a fresh product and you're very afraid that your consumers are going to find infestation, you'll probably set a pretty low threshold in terms of the number of males you're catching in your traps for when you trigger. But yeah, it's a sliding scale basically. So as you move up in terms of the average number of males you trap, that's when you're going to have a higher probability that you're going to have infestation in the fruit. But I would stress that you do still need to trap. So it's good to trap, know what's going on in your field. I've seen some sites where we might trap them, 2 miles away, and other sites where we might not trap them for a couple of weeks later. So there's landscape effects going on. Some traps are, they have a greater wood edge with more non-target hosts out there. So that's another thing I didn't touch on, that there's a lot of things that spotted Drosophila will build up their populations on. So some sites might have higher non-target or non-crop hosts out in those wood edges that will affect basically the populations around there. So trapping at your own site is important. Hopefully that answered the question.
Feel free to shout out a question if you have it.
Is that my Yes, Richard.
I burn my field, and for a number of years I mowed it. I did it mechanically. I would assume that burning helps with the pest infestation, but how does it compare to mechanically mowing it?
Yeah, so burning will definitely probably help with your other pests, your spanworm, etc. It might help a little bit with your fruit fly as well. Spotted Drosophila, we don't really know. They don't have we don't know where they overwinter. So I suspect even if you burn your field, they'll be overwintering in the woodlots around there and stuff. They're really mobile compared to some of our other pests. So yeah. I don't think that would impact them as much.
Okay, I just wondered how one was compared to the other.
It is a good question because we haven't specifically monitored, or I haven't. I'm not sure if my predecessor had monitored at sites that traditionally burned more versus mowed more, but I suspect given how mobile the pest is and how much they move around, that it wouldn't impact it too much.
I do have a slightly wet area on one end of my blueberry field too, so I'm sure there's some coming from in there.
Yes, I mean, really damp areas, they can kind of hang out in those areas quite a bit because they love that humidity.
Okay, I'll let somebody else get in here. Thank you.
It'd be a good area to hang your trap.
Okay.
I wonder, Phil, what you think about SWD and having fields on one cycle rather than having a split field. Like for blueberry maggot fly, that's a trick that works pretty well.
Yeah, again, given they're so mobile and there's all of these crop hosts. Even, you know, in the off cycle year, the prune year, I mean, they'll be out there on fungi. We've got them to develop through on pure maple syrup. We've got them to develop on pigeon poop. They're really, you know, they're ferocious. They will develop on anything. So their populations, that's why their populations can grow quite fast, because they see everything as a potential host, this species, which makes them so different from blueberry maggot fly. So hopefully if we eventually do get a release for this biologic control agent, I suspect I'll have a good number of people who let me release it at their farm. Right?
I'm sure you'll have plenty of takers.
Well, we're hopeful that they'll be very effective and help reduce this fruit flies population.
Any other questions?
Does the amount of ripeness have an effect on when we should begin trapping? The amount of color, blue?
Yeah, it's a good question, Paul. So I would trap, you know, just as our fruit was gaining some color. One thing we've also noticed is as our fruit gets riper, so as it gets darker, it starts competing with our traps. So our trap doesn't catch as much as our crop comes into ripeness. So really you want to get them out early to sample that population. But as our ripeness increases, you know, if we were 100% ripe, our spotting would probably wouldn't go to our trap as much, because, you know, that's a red piece of plastic versus the crop, which is, you know, a beautiful, nice wild blueberry smelling really good, covered in yeast, which they really like. So I would trap just as the color comes on. But one caveat is that as our crop gets riper, it's probably taking away or attracting the fruit away from our traps a little bit. Yeah, that's a slide that I should add into a future talk, just showing how our traps decrease in terms of effectiveness as our crop gets riper. So there is a big competition between our traps and our crop.
So I discovered fields that I spray for the regular blueberry fruit maggot, because I'm putting the insecticide out for the blueberry fruit maggot. That delays when I will find the SWD in my traps.
Yeah. And do you usually do a perimeter spray for your blueberry maggot?
Yes.
Yeah. So I guess I theorized that some of those early perimeter sprays are definitely helping with our spotted Drosophila infestation, given we're seeing that edge effect now more strong earlier on, particularly. Yeah. And a lot of the same insecticides that will target blueberry maggot will also target spotted Drosophila for sure. Well, I'm happy to stay on for as long as you folks have questions from me. So
I have another one.
Oh, great.
We need a permit or a license to spray for these maggots?
Obviously, you need your applicator's license through the Board of Pesticides. But other than that, you can make applications just like you would for blueberry maggot fly.
Okay, I have a lot of wild apple trees too on the property. It used to be managed for apple products. The flies go back and forth, do they?
So they don't attack, you know, ripening or ripe apples. What we found is late on in the season, when, you know, we've harvested our fruit or, you know, our blueberries, if there's dropped fruit, particularly apples, actually, because we found them in apples, they'll use them as a sugar source. So they'll go to that. There was a great study done in Michigan when I was there by Harit Ball, and she picked up fruits from all of the other orchard crops around Michigan, and she could rear spotted Drosophila out of pretty much all of them. So if you have dropped or downed apples, they will definitely use them as an overwintering source. Mm-hmm. One of the things we were doing that experiment was, because obviously an apple is a little bit more robust than a blueberry. Usually they don't dry out as fast, so we were thinking that they would overwinter for longer on, you know, a big fruit such as an apple. So definitely having an apple underneath a snow cover, you'd have a lot of resources for them to survive out the winter.
Mm-hmm. Okay.
You might also see some higher numbers around that apple orchard because of that.
I normally keep a lot of my apples that are around the field raked up, and I feed them to the pigs or the chickens or whatever. But
yeah,
I just wondered if the larvae would actually go back and forth in the winter.
Yeah, definitely when they drop and get damaged. Once they're damaged,
mm-hmm.
the skin is broken, the spotting will get in there.
Okay, just like yellow jackets then. Thank you.
Yeah, unfortunately. So the sanitation in and around the field can be really key, especially in smaller setups where you might have multiple crops.
There's a chat question. How often do you change the fluid in the traps?
We change them weekly. So that's probably because of, you know, just driving out to sites from here in Orono. Obviously, if you check them more regularly, you get more regularly up-to-date information. Checking them more regularly too would probably mean that they're not as smelly when you go back to check them. So obviously those yeast sugar traps, if you leave them out for a week, they usually smell pretty bready after a week. So if you were changing it over more regularly, you probably would avoid a lot of that smelliness, and you probably get more up-to-date data. So yeah, we check them every weekly just because of all of the driving.
The question I know you get a lot, and I hear it once in a while, is can I use kombucha or some other juice, fermented juice, as the trap liquid?
Yeah, they really like fermentation volatiles, so that's why the yeast and sugar work so well. But I think they will go to any other juices. I've seen traps with, you know, apple juice and apple cider vinegar mixed together and stuff. They will go to pretty much anything. I mean, they're mostly changed in the sugars, but really fermentation volatiles are a big cue for them, so they really go to that. There is another really good bait spray that has been released on the market in the United Kingdom. I'm not sure. They've done some trials of it here in the United States, and my lab is hopefully planning to do some trials on it this coming season if we can get it. But it's based on a yeast, Hechuvarum, and yeah, H is the genus name, Hechuvarum, which is this yeast that's commonly found on grapes, and it is the most attractive yeast you can find for spotted Drosophila. But they've combined that with strawberry juice and some plant proteins to basically come out with this new bait spray. They also claim it works on fruit flies, but they don't have any data on blueberry maggot. But it is one that I'm hopeful my lab will be able to test this year as well. But they'll be attracted to all juices. I should try answer my questions a little bit shorter. Me and my tangents.
Okay, I think that's about it. Thanks so much.
One more question. When you're doing the salt water, I noticed the slide, you do not crush the fruit. So crushing the fruit's not important with the salt water?
So our original recommendations were to crush the fruit. We think if you leave them long enough, it doesn't matter as much. I think previously we used to crush it a lot, and you'd have a whole load of seeds in there, and they used to get very messy, and it was hard to see. This year we basically, as we would rake the berries, as we were putting them into the salt water, we just make sure that there was no stem scar or stems left in the stem scar, because they can escape out the stem scar. But we would also, maybe give it a little bit of a crush, but not mash it like we had previously done. So it does help to maybe give it a squeeze, but not to mash it down, because what we found is when we mashed it a lot, we had a lot of gunk, and it was harder to see the larvae. But so it's not overly necessary, but it could help if you maybe just pinch them a little bit, just a tiny bit, but not squashing them. Usually when we rake them, we do enough damage to detach that stem scar or the stem, so that they can escape out the stem scar if they're sitting in the salt water.
I had a question. Yeah. Do you know if they have any preferential environment in which they overwinter in, or are they pretty generic in their choice of habitat?
So when we've always done our studies for overwintering, we put them in leaf litter in the edge of the crop, in and around, we'll say, the tree line, just because when we've previously done a lot of trapping studies to look at their phenology and stuff, we find that late in the season they go into wooded areas, and that's where they're kind of hanging out late in the season. So we think that they overwinter in the wooded areas in the leaf litter down low.
Has there been any thought about doing early spraying in those margins or that habitat to see if it has any impact on the hatch or the migration into the field?
I haven't seen any of those studies, and we haven't done any here, mostly because we come up against the issue of spraying off the crop. There's a lot of, obviously, our insecticides aren't labeled for just being spraying into the woods or on the woodlot. So that we haven't tested that here in Maine at all, no.
Okay. What about biologicals? Has there been any success with any of the biologicals as far as trying to control the population once it's established?
When you talk about biologicals, are you talking about BT and
systemic stuff, something that they would ingest that would affect their reproductive capacity, or something that would knock the population down endemically, rather than just kill the individual bug, but something that they would ingest?
Yeah, so I know there's been a lot of work done on Beauveria bassiana. Look at that. I think the issue with it is they do come in contact with you, and it is effective, but it takes a long time to kill them off. And in that time, usually they're still infesting the fruit. So I think they do work, but they just don't work fast enough to basically, impact the population. One thing about, obviously, spawning Drosophila is it takes a week for them to develop from egg to, or maybe a little bit more than a week, 8 to 10 days to go from an egg to a female that's ovipositing or laying eggs.
Right.
So it's a very fast generational turnover. So that's why their population can build up very fast. So when you have a Beauveria that doesn't kill them for 5 to 6 days, they can have done a lot of egg laying in that period of time. So.
Okay, thanks.
Very good.
That triggered a question. How long does an adult female live to lay eggs?
Yeah, so This is probably confounded a little bit because we bring them into the field or into the laboratory to do a lot of these studies. So we bring them in and we put them into a nice, container with diet and a really nice climate. But for the most part, they could live for up to 21 days. And we expect each female to lay about 350 to 500 eggs in her life cycle, if you know what I mean. So they can live for quite a while, and they can, unfortunately, lay a lot of eggs. So that's again why they're so destructive and why we're doing so much research. So our traditional blueberry maggot fly would, they come out in a pretty tight emergence. We only have one generation of them per year, and we can control them pretty effectively based on our degree days and our traps and a few targeted sprays. Whereas spotted-winged Drosophila, once they start, they're just going through multiple generations, and for the most part, they keep going till they either freeze to death or they go into that winter morph stage and overwinter in the area. The majority of them do freeze to death, though. So when we think about insecticide resistance, etc., for the most part, every winter here in Maine, most of our population probably dies, over the winter. But it only takes a really small amount of them to survive to restart the population the next year. So when you think of that 350 to 500 egg range, and if it's warm enough that they develop so fast, it really doesn't take long for their population to build up pretty fast.
I'm back.
That's good. That's good.
I manage about an acre of wild blueberries. It could be more if I cut the woods back. I think the whole property would be blueberries. I have a neighbor up the road from me. He's fairly close, probably 200-300 yards, and he has all wild blueberries. The whole hillside and behind his house is all wild. If I treat mine, is it going to be a losing cause if he doesn't treat his?
So for the most part, when we treat, a lot of it is about, putting insecticides onto that fruit. So when they, land on your fruit, they die basically. So the population is can be so large in and around the woodlots and all of those areas, that regardless of whether your neighbor treats, it probably wouldn't matter if, wouldn't matter if he doesn't treat, if you treat yours, because they're just not going to be using his crop as a host. They're going to be using multiple other things in and
Okay,
around.
so it still would be worth my while.
If you were, if you were finding high numbers in your fruit, or if you were having high infestation and you were trying to market it fresh and you were concerned that people were, finding larvae in there, it will be probably beneficial to treat.
Okay.
My hope would be, our biological control agents will eventually mitigate that need, but we won't know that for multiple years and lots of research.
Mm-hmm. Okay. All right. Thanks again.
No worries.
From that conversation, part of what I say, my blueberry field could be attacked by SWD coming from a different crop, even like raspberry. Correct?
Yeah. So, I mean, I know Frank had done a lot of work looking at all of the things that grow outside of our crop that they would be a host on, and it's a pretty large list of things that they'll develop on, or fruits in the wild. But yeah, raspberries too. If you have wild raspberry or bramble that are untreated, the spotted-winged Drosophila can generally find them and oviposit into that fruit.
So the bottom line is we don't have to think that SWD on my field does not have to come from another blueberry field.
Yeah, no, don't.
Hey, Phil,
Yes.
it looks like on your coffee filter, there may be some blueberry fruit fly larvae there. Can you use this new test to test for larvae from the blueberry fruit fly also?
Yeah, we do count blueberry fruit fly in there as well. There is a great, maybe I can put it in the chat real quick. There is a great paper, and I should be able to get it, and you should be able to download it. I'll stop sharing my screen so we can all see each other. See if I can pull this up. This should give you more information on how to how to do that salt test. But it also has some nice pictures of the difference between spotted Drosophila and the blueberry fruit fly, and how to tell them apart in your sample. So let me find this quickly. Bear with me.
While Phil's looking for that, I just have a few people who I'm not sure who you are. So if you could just say, if Fire Tablet would like a pesticide credit, if you could just let me know or put your number in the chat, that'd be good.
Yeah, hey Lily,
And
this
then-
is probably Carrie Nash on the Fire Tablet. I changed from
Oh,
the
okay.
computer to a tablet.
Okay, great. I got you. You're on the list.
Yeah.
And then the other one is a number, which is MS9745. Anybody?
Hi, that's
Lily,
me. I'm just observing.
Okay, no problem. Thanks for joining.
Okay, so I just put a PDF into the chat. So if you click on chat, I think you can click on that and you should be able to download it. And in there, let me share my screen again. We can look at it. You can see the difference, Carrie, between the different types of fruit flies. So share. This is a scientific paper, so there's a lot of boring stuff in here, but it does go through some of these methods. So here's your different sizes of spotted wing Drosophila: an egg, a larvae, second instar larvae, and these are the big third instar larvae that we really don't want to see because they're the ones that are most visible and they do a lot of damage to your fruit.
Was that picture under a microscope, Phil, or was that wasn't just a magnifying glass, was it? Magnifying
This one
glass?
here, I suspect, was a microscope, yeah.
Yeah,
So,
okay.
I think we put
Thanks.
in for scale 5 mils. These ones would be, 3 to 4 mils, so they get pretty big, unfortunately. Let's see if we have a. So we had a picture showing the difference. Maybe we didn't. Well, these are the different instars. For the most part, you'll see the difference between the spotted wing Drosophila, because the spotted wing Drosophila have these kind of what looks like a tail, whereas the blueberry maggot fly are a little bit rounded and they have 3 yellow stripes on their butt, basically. Excusing my language. So you can tell the difference between them. They also move a little bit different, but maybe we can get I can get a picture, and I'll either email it to you, Carrie, or maybe we can update it on the UMaine website so you can tell the difference. But you can definitely tell the difference between them. It might take a little bit of a better eyesight and that a strong hand lens, but particularly in the larger larvae, you can tell the difference. I'll stop sharing that. You probably don't want to look at those fellows a lot. They're not that good to look at. Unfortunately, Judy Collins looks at a lot of them. If she's still on this call, she's probably laughing.
Extra protein, right? That's what they say.
Yeah, yeah. I think I've probably eaten a lot of them in my time working on this species. I do have a tendency to sample the fruit while I'm out there, unfortunately.
Okay.
Yeah, well, thanks for you all joining. I see somebody's going off to shovel. Best of luck with that. It's quite the dump of snow. Yeah, thanks for joining and all the great questions from everyone tonight.
I did, I started recording a little late, but we do have a majority of the presentation. So we'll have that edited a little bit and then up on the website. So yeah, thanks for joining. Have a great night.
You'll see me back on March 5th where we're going to be talking bees, I think, right?
Yeah, that'll be great too. And Friday is weed control. And it'll be a good talk because it's somebody from New Jersey at the Rutgers High Bush Blueberry team. So
All right.
hope to see them.
Lily, this is Paul Cleveland. I wanted to make sure I was on your list.
You are. I got you, Paul. Thanks.
Thank you.
Yep, no problem. Take care. Bye-bye.
Thanks, Paul.